So if you listen to our last microbiome episode, you know now that it's extremely important to your health and you've got questions. Luckily, we've got answers. Since our last podcast, we've actually added an amazing PhD microbiologist to our team. Dr. Eric Battaglioli comes to us from Mayo, where he was doing research on the microbiome in the context of individualized medicine, which is exactly what we're trying to practice. We've been blown away by conversations with him, and we want to share his incredible knowledge with you as well. We're going to answer some questions we received and just dig deeper into the microbiome in this episode, which now that I say it out loud sounds totally disgusting.
Hey there, I'm Mike.
And I'm Matt.
And I'm Jody. The Wild Health Podcast is our quest into human performance, longevity, and personalized medicine. Together, along with experts in these fields, we'll learn how to live longer, better, and become smarter and stronger. We'll do the deep dive into the research and hard work of putting it all together, and you reap the benefits.
Dr. Battaglioli, the other day we were chatting about the microbiome, and I thought we had been talking for about 15 minutes and it had been almost 2 hours. Your knowledge on the matter just blew me away. And honestly, I wish we had recorded it, but we didn't. Luckily, it was interesting enough that I'm really stoked to actually talk about all that stuff again and record it this time for other people. So could you start by just describing some of your research and your interest related to the microbiome?
Sure. So my formal training is in basic microbiology and bacterial pathogenesis. So I started out my research career interested in what the types of ways that microbes use to make you sick and trying to figure out methods to combat that so people don't get sick with those infections. And that took me through a bunch of meandering paths. And towards the tail end of my graduate work and into my early postdoc days, I started getting interested in not just how microbes make you sick, but how they communicate to and with the host to create some type of interplay that creates outcomes that are important for our health and well-being. And that led me to microbiome. So I spent some time in looking for labs that I could join to work on those areas. And I wound up at Mayo Clinic investigating how the microbiome interacts with pathogens and creates structures that can lead to conditions that we describe as dysbiosis, which can leave us susceptible to infectious disease with things like Clostridium difficile. So my work at Mayo Clinic was targeted around trying to understand how real patients may be susceptible to infection and using animal models to work with us to understand how a person can get sick and not just contrived models or animal models of infection.
Yeah, so I try to read all the research I can find on the microbiome, anything I can get my hands on, because it's so fascinating. But you're actually one of the people who was producing some of that research. So you've got answers to a lot of the questions that I have when I read this research. And Mike's not with us right now. He's probably going to be joining later. He's probably going to have some questions as well. But I've got so many, I'm just going to start firing them at you. I think a good place to start would be to basically say, just for a baseline, what are your top 3 actions somebody could take to optimize their microbiome?
That's a great question. As we dive deeper into understanding the microbiome, we're appreciating that essentially everything we do can affect our microbial community structure and function, down to what we eat, where we live, how much sleep we get in a given day, who we interact with on a day-to-day basis, if we have pets. Everything we do affects our microbiome in some way. So I think as a first level, just being cognizant that all of our actions matter and that even small things can, when done in parallel, can add up to have effects that can be lasting is really important. So being cognizant that what you do has effects is important. The second biggest thing I think is being cognizant of your diet and what you eat. You're not just feeding yourself when you eat, you're feeding the microbes that live in and on you, and what you're able to metabolize and to eat.
It looks like we've got a caller. Let me answer this. I think Mike's calling. Caller number 1, you're on the air. Dr. Eric Battaglioli is with us today. Who am I speaking with? Do you have some questions for the esteemed doctor?
I do. My name is Mike. I hear that Dr. Eric is the colon whisperer. Is that true?
I don't know if I've ever been described as a colon whisperer, but I've certainly seen my fair share of mouse colons over the years.
So we got off to a great start. Thanks for ruining it, Mike. Just to summarize where you're at, Dr. Battaglioli, not Dr. Eric, which is very disrespectful, was just telling us his top 3 actions to optimize the microbiome. And just to summarize, because I know you've had this question, sounds like location is really important. And then food, we're getting into food and what you eat and feeding your microbiome. So thank you for the interruption. If you don't mind to mute and just listen in, that'd be great from here on out.
I can definitely mute it. Could you just expand on what location means? I'm not sure I exactly understand that.
Yeah, it's a good question. So Eric was talking about where you live and things like that. What do you think, Eric? Are there specific action items when it comes to getting in nature more, or where you live or how you live when it comes to the location?
Absolutely. I think in general, a lot of people ascribe to the idea that cleanliness is key and that we should be living in the cleanest possible environment that we possibly can. And I'm not sure that I agree with that. Your exposure to microbes is important, and you need to have access to a diversity of microbes to help build a complex and diverse microbial community. And that's important for keeping you safe and healthy, make sure your immune system is working properly, and make sure that your communities are stable and robust that can protect you from things like opportunistic infections. So living in a bubble isn't necessarily a good thing. Purell has its place, and there are times when it pays to be clean, and there are environments when it pays to be clean when you're in those places. But generally, getting outside is a good thing, and exposing yourself to the world around you is a good thing for your microbiome in moderation and when appropriate.
I was feeding my 1-year-old some scrambled eggs this morning, and she dropped some out of her chair onto the floor, and I picked it back up and put it back on the tray. And my 7-year-old said, "What are you doing? You can't feed her that." I said, "She just enhanced those scrambled eggs just now." Of course, she just looked at me and then turned around and go in and told mommy on me. But I do think that the 3-second rule should maybe expanded to the 10-second rule or the 5-minute rule. And yeah, I think we definitely are probably harming ourselves with washing all the times. We talked about that a little bit in the last podcast we did. We also talked about nutrition in general. Do you want to expand on that? What kind of exact things you can do to enhance your microbiome when it comes to the food you eat?
Absolutely. So the majority of the microbes that live in your gut are in your colon, and the majority of our nutrient uptake happens much sooner in the GI tract than that. So we extract most of the things that we need out of our food as it passes through the small intestine. So the things that we can't digest or get access to are most of the things that do get access to our colon and feed our microbes. So things like non-fermentable fibers, inulins, oligosaccharides, things that our digestion is not equipped to be able to process. That's a five-course meal for the microbes that live in our colon, and that's what they really need to flourish and to survive. So eating foods that are rich in those non-fermentable fibers is key to being able to feed your microbiome.
Yeah, I think we all know how important it is to eat our vegetables. I think we don't understand most of the time exactly why. There are a lot of micronutrients and vitamins, but I think probably as or more important is just that, feeding those good gut bacteria. Do you have a third item, or do you think those two really are the two most important things to think about?
I think one of the other more important things that you can think about for optimizing your microbiome is being aware of the things that can cause insults to your microbial structure. So our bodies are remarkably resilient, and our microbiome is remarkably resilient. It responds to perturbations with great resilience, and it can bounce back from a lot of things. But I think it's important for us to all appreciate the types of insults that can cause us to stress or destabilize these structures, which can lead to problems downstream. And I'm talking about things like antibiotics or chronic conditions like not sleeping well, or dietary shifts that maybe are not the best thing out there for your microbiome. Those things can add up to lead to pushing your microbiome in directions that are bad for you over the long run. So as an action item, being aware of what can disrupt your microbial communities and then trying to limit or avoid those activities.
So last time we did mention a little bit about sleep. I think that's important. And then as far as the things that harm, we know antibiotics, we've talked about that. Specific nutrients. I did read a study just recently about sugar and how harmful that is for the microbiome, more than just the spikes in your glucose and your insulin sensitivity and what it does to that. So not only feed your microbiome the correct things, but don't hurt it. Chemicals like antibiotics and sugar. So what about testing? That's a question that we've gotten quite a bit. How much can you test right now?
So that's actually one of the great things about microbiome research these days. And I think one of the things that has fueled the explosion in microbiome research recently is that the tools that are available to study the microbiome are now more accessible and more available than they've ever been. Even 5 or 10 years ago, the tools and techniques were still around, but they weren't as good and they were incredibly expensive. So really only academic specialists in this area had access to these kinds of techniques. And we're now seeing, as a result of improvements in the sequencing technology and the ways that we can look into the microbiome, becoming consumer accessible because they're cheap enough for us to be able to go after and do for your average everyday person. So the tools are getting a lot better than they used to be. We have more sensitivity. We can do a lot more for a lot less money. So to answer your question of, what can we test for right now, we can look very deeply at the composition of the microbial community. So who is there in a particular sample? And we can also look at what they're doing. So what genes are these microbes expressing at any given time, or metabolically, what are they producing and how are they speaking to one another and how are they speaking to the host and how is the host speaking back?
So in our next podcast, we're going to be interviewing the Chief Science Officer of Viome, which is a company that I've used. Do you know anything about their tests? Like what type of tests do they do? And what other types of tests are there out there?
Yeah, so it sort of depends on the particular company as to what technology they ascribe to. Some choose to use what's called 16S ribosomal RNA sequencing. That's a lot of very big words to say that we are looking for little tags or barcodes that are common to most microbes, and the sequence of that tag or barcode tells us what microbe it is. So we can look for and sequence that tag in lots of different microbes all at once in one sample and say something about what the population is. So it's a very powerful tool. The downside is there's very limited resolution. We can sort of identify some things, but we miss a lot of other stuff. And the taxonomic resolution, our ability to say this is a particular strain or a particular species, is very limited with that technology. Going a bit deeper, which my understanding of what Viome and a few other companies use, is a technique called shallow metagenomic sequencing. And in this technique, instead of looking for one particular gene that all microbes have, we just sequence everything that we can out of a microbiome sample, all of the microbial DNA that's there. And by sequencing everything and comparing all of those terabases of information, of genetic sequence information, to established databases, we can more precisely identify the types of microbes that are there instead of using a single common gene. And that's a very powerful approach as well, and it gives us much finer taxonomic resolution about who's there in a particular sample.
Yeah, I think they talk specifically about metatranscriptomics. So what would be the advantages versus disadvantages of that technique?
Yeah, so metatranscriptomics is then a further adaptation of the metagenomic approaches where you're looking to sequence all of the RNA that's in a sample. So you're asking not just who's there, but also what genes are they expressing at any given time. That's another layer of information, and in my opinion, arguably much more valuable, because you're not just concerned with who the members of the community are, but what they're doing and what activity they have. And as we begin to unravel the microbiome more, we're beginning to appreciate that it's less important who's there and it's more important what they're doing. And part of the reason why there might be such great variability in microbiome structure, even amongst people who are healthy or genetically similar, is that they have diverse microbial communities which are functionally equivalent. So they have different microbes performing the same tasks. And that is very hard to resolve when you're just looking at composition.
So how would you use that information that you got from a company like Viome?
That's the $64 million question, I think, is how do you interpret the activity of my specific microbes and translate that into something that's meaningful for the person to direct their health and well-being. And we're just starting to scratch the surface of that question, I think, because I like to describe this as a stool with 3 legs on it. There's 3 things we have to consider simultaneously. There's the microbes they have and what they're doing. There's the host genetic background, the person who we're studying or the patient, and then the environment that all of that exists within, both the microbes and the person. And we can't really make we can't make real precise recommendations without considering all 3 of those things simultaneously, because they all work together to create one contiguous output.
So it sounds similar to when we get lab testing for someone or we get genomics testing for someone. It's hard to make recommendations based just on one of the two. We want to look at both and kind of the interplay and also the person's life in general. So do you have an example of when someone's genomics and their microbiome and their lifestyle, you kind of put those together to make a recommendation?
Yeah, so it's very hard to do because there's so much variability in host genetic backgrounds in conjunction with microbiome structures that we're struggling to find enough data to be able to draw definitive links between specific genes in a person and specific types of microbes and their activity. So it really comes down to a case-by-case basis. But what the literature is suggesting is that there's very clear interactions between the activities of microbes and the genetic backgrounds of hosts. And there's complex language and complex discussions that happen between those two groups. So it becomes challenging from a consumer perspective. If you get a report back that says, I have these types of microbes in my gut, I should do X or Y. That's a bit difficult to interpret. The question becomes, well, how do those types of microbes interact with my genetics? And what should I have my microbes do or try and encourage my microbes to do in order to benefit my specific host genetic background? And the data and the jury for that is still out, I think.
Yeah, when I got my Viome results back, I remember one thing that stood out to me is they recommended I stop eating spinach for a while because there was some sort of spinach virus that was found in my gut microbiome. But I know the benefits of eating spinach. And so it was kind of for me, like, you know what, I'm not having any issues right now. If I was having gut issues, maybe this would be good information to try, but I'm not. I feel healthy and I feel good, and I'm not going to stop eating spinach. So I think getting a recommendation like that from Viome without knowing the person and whether they're having actually any dysbiosis or any problems, I think you really got to take that into consideration, what the person is clinically doing and how their symptoms are now before you can really give a hard recommendation just based on looking at someone's stool and a microbiome. And we'll put Viome on the spot. I mean, you have no incentives here one way or the other. They have a product. So we'll make them tell us how exactly they're using this data and how much they think you can take actions from their test results. Kids, this is a really big topic. A lot of people have, like, how can I help my kids' microbiome? We know how important it is. I saw a study recently on obese kids and teenagers and how different their microbiome was. And whether that's an association or there's causation, we have no idea. But we know this is important. You know, we know the germ-free mice studies that we talked about in the last episode. So there's probably something there. What can we do for our kids to improve their microbiome? And I guess a specific question before we talk about that is, do we have a specific window of time to get our microbiome right? You and I were discussing that before, and you were telling me some interesting data on that.
Yeah. So as a caveat, I'm a parent too, and I have a kiddo that's in this window. I have a 16-month-old at home. So this is something I'm thinking about all the time in the establishment of his microbial community and doing the best that we can to give him the healthiest microbiome possible. The window is about 3 years. So from the time of birth to about age 3, that's when your microbial community is going to suss itself out and start to establish its structure into what it will become for the rest of your life. Once age 3, you hit age 3, more or less what your community structure is, it's going to remain pretty similar to that for the rest of your life. And that's in the face of antibiotic exposure or travel or moving to different parts of the country or even changing your diet in significant ways. The bulk of what you have at age 3 is what you're going to carry with you for the rest of your life. So if there's a fragile point in your microbiome development, it's really those first 3 years, and making sure that you're exposed to the right kinds of microbes during that time is really important. And also trying to limit your exposure to things that can disrupt that process, like antibiotics.
Yeah, and it's so scary to me. I see so many kids, how many courses of antibiotics they get when they're 1 and 2 and 3 for ear infections, which is almost always completely unnecessary. Strep throat, which is usually unnecessary. Any kind of viral infection, they just get written an antibiotic prescription. And it worries me so much. I've got 4 kids. One of my children, when she was 6 months old, was in the ICU. And I remember so specifically, she was actually pretty sick. She was intubated at the time. It was a scary time. And she didn't have a bacterial infection, but she spiked a fever one day, which you can get just from being intubated. And I remember them talking about putting her on IV antibiotics, and I just resisted it so badly because we had no clear source. It was probably just related to being in the ICU and being intubated. And I didn't really push against them too much. I kind of mentioned at first, but in the end, I ended up caving and letting them start, even though I didn't think the literature was supportive, but they're the PICU doctors and I'm not. And then that child of our 4 has had a lot of gut issues and other things that every time she vomits, I think, did I mess up by not pushing back against antibiotics when she was 6 months old. And for my other children, my 2 youngest, the 2-year-old and the 1-year-old, have neither one ever had a course of antibiotics. And there's times they've had fevers and things and we could have given it to them, but I just think it's so important to really protect that microbiome of our kid when they're young. In our last episode, we talked about important things like vaginal delivery when possible, breastfeeding when possible, playing outside and playing with pets. Are there any other things you do for your kid or certain things you look out for?
We definitely do all of those things. And we're always thinking about trying to make sure that he's getting as much reasonable exposure to safe microbes as possible. So we have about the 10-second rule in our house as well. And some days it seems more like the 10-minute or the few hour rule. If there's something that we were feeding him for breakfast that we missed on the kitchen floor, and sometime in the afternoon, we catch him under the kitchen table snacking on a Cheerio that's been there for a few hours or days. And I don't worry about that. I think it's okay. Our house is otherwise free of the stuff that I'm really worried about him being exposed to. So that's a good vector for being exposed to good microbes and to help build that. To dovetail on your point of, other things that you can do and the antibiotic thing, be an informed parent, ask questions, push your provider. Do you really think my kid needs antibiotics? Is there a self-limiting aspect to this? And especially the ear infections. The vast majority of ear infections are viral based, but we still provide antibiotic treatment for that. And should we be really doing that? So judicious use of antibiotics is really important. And also what we're feeding our kids. We go out of our way to try and find the best possible things to feed our kid. And that includes paying obscene amounts of money for antibiotic and hormone-free milk and antibiotic and hormone-free products. And that's an aspect that we sometimes overlook, that the acceptable levels of antibiotics in food products is reasonably high. There's detectable levels of antibiotics in food products that are given to livestock and used on crops and things like that. And we can detect those things in our food sources. And there's evidence that low dose antibiotic exposure over long periods of time does affect microbiome structure. And it can affect a wide variety of different long-term chronic conditions. And I've actually heard some investigators suggest that part of the reason why the human race is increasing in size and mass and bone density over generational time is the same mechanisms and reasons why we provide antibiotics to our livestock, and that the reasons we feed tetracycline or other antibiotics to livestock to make their mass increase more quickly is having the same effect on us because we're drinking that milk, eating that beef, eating those eggs.
Yeah, and you talked about the quality of food. I know it's more expensive to buy organic, but even non-antibiotics, but like glyphosate has been shown to have negative effects on the microbiome. So when you buy vegetables at the store, you feel like you're doing a great thing, but if those have that residue on it, I mean, that could definitely be causing some harm. And you made a great point about pushing your provider as well. As a physician, I can tell you, when you've got a kid in front of you who has an earache and a fever, you got a decision to make there. You can pull out the prescription pad and write a prescription for antibiotics and you're done. The visit's over. Or you can spend 5 more minutes actually talking to the parent and telling them the harms and the benefits. And when you're really pressed for time, in the way the incentives work in our medical system, you're going to have to push the provider or they're going to take that 5-minute shortcut a lot of times. So it really is incumbent on you to protect your children's microbiome. How hard is it to change their microbiome later if they didn't have a great first 3 years, had chronic infections and other issues?
It's tough. Your microbiome, once it establishes itself and forms a complete community where all the various ecological niches in your gut are filled, it's hard to move it around in a gross fashion later on in life. I like to tell folks when we're talking about this type of thing that you can give it nudges in different directions. Sometimes you can get probiotics to stick Not everyone is a probiotic acceptor, and they require continual application of the probiotic essentially indefinitely and to keep that microbe present in their community. Others are a bit more accepting. We don't really know all the interplay of what makes someone a good acceptor versus not, but it's hard. It's hard to make global large-scale shifts in microbiome structure once you pass that 3-year mark. So that's sort of the critical window of, I want to be thinking about what types of things are my kids exposed to? What am I feeding them? Are they getting good exposure to microbes throughout that important time?
So speaking of probiotics, in the past, I've believed that's a great thing to do right after a course of antibiotics, help someone reestablish their microbiome. But there was a study in Cell that came out, I think in September 2018, where they actually looked at people after a course of antibiotics. When they gave them probiotics, it actually halted the regrowth of their microbiome. And it makes sense. You've got thousands of microbes and you're giving them 3 different species with a probiotic. And it's a very competitive world down there in the microbiome. And in this study, they showed that by giving them those 3, it kind of stopped all the other microbes from coming back. And it seemed to cause a little bit of harm. I mean, since that study, I don't know how I feel. I think, honestly, if it's my kids now after reading that study, I'm not going to give them the probiotic. What are your thoughts on that?
Yeah, I think it depends on, what's your goal? What's the outcome that you want to create as a result of giving this probiotic? And is this a species that's normally present in this person's microbial community? Or is this something that you're trying to add after the fact? Like I said, the microbiome is generally a pretty stable thing once you hit age 3. And even after you take an antibiotic course in the days to weeks to months following treatment, your microbiome will repopulate to essentially what it was before the exposure, which is not to say that the acute exposure is not a problem and doesn't create issues that we may have to rectify downstream. But your body is, like I said before, your body's really good at finding homeostasis and very good at establishing a stable baseline that it can return to. And if we push too hard on one particular direction or another without really knowing what the effect of that particular microbe and that particular person will be, it can lead to some of those off-target effects.
Yeah, and I think it's important to note that even though in that study the microbiome did return after I think it was a few weeks, it actually got back to the normal microbiome. That's not to say that those antibiotics weren't harmful. I saw one study where the risk of anxiety and depression after one course of antibiotics over the next 12 months would increase 17 and 24% respectively. And if you got 2 courses of antibiotics, then your risk of anxiety and depression were up 40 and 45% over your baseline risk, which is pretty scary. So even though your microbiome bounces back, there are definitely problems associated with that. So getting your microbiome back to normal quickly, I think, is important. Let me tell you my thought on this now, and I want to get your take on it. If I have to take a course of antibiotics right now, based on my reading of the literature, one thing that I would consider, and this is kind of out there, I don't know if anyone's actually doing this right now, would be that I would probably, before I took the antibiotic, save some of my poop, freeze it, and then consider doing an enema right after the course of antibiotics to get my gut microbiome back to where it was more quickly. I saw a study in cancer patients, at least, where that worked. It seemed to be very safe and effective. I haven't really seen a lot of other data. How do you feel about that?
That's a good question. So people have been doing sort of the home microbiome reprogramming for some time, which, as a pathogenic bacteriologist by training and a microbiome trained scientist, I'm not sure I can recommend for your average person in their kitchen. But there is data, as you say, and I saw that paper too in Science Translational Medicine, and it was a really well done study which showed a nice repopulation effect, an accelerated repopulation effect by doing this autologous transplant after the antibiotic exposure. I think there's potential there. The trouble is I don't know in the average everyday population how well that's going to stick. So the patient population that they looked at in that study was individuals that were receiving stem cell replacement therapy. So they already had a number of confounding variables and some medical conditions that may have an effect on their ability to receive or to accept the graft better or quicker or differently than your average person with your average course of antibiotics may. So it may be a little challenging to translate those data to your average patient population taking antibiotics for your average infection. I definitely think there's potential there though. And those are experiments that I think we should do, and we should, utilize the germ-free mouse and some other models to try and explore those possibilities.
So you're a scientist and you don't like to make a recommendation unless you have clear data. But let me tell you the problem I've got. I see patients and sometimes they have to take antibiotics. What I would like to offer to them is to say, hey, if you want me to free some of your poop and then give you an enema afterwards, I think that's probably going to be beneficial for you. What do you think are the potential harms or downsides to that?
That's another great question. And yep, I am a scientist and I live my life by the data. So aside from the procedural aspects, whatever risks are associated with the procedure itself, I think a consideration is what did their microbial community structure look like before they took the antibiotics? And was it optimal to begin with? So should we be pushing them towards the same microbial community structure after an insult, or should we be taking the opportunity to move them towards a different structure? And that raises a whole series of other questions of how do we define what's good, and good in the context of that particular person, because good for me is not necessarily going to be good for you or good for someone else. So yeah, we should be thinking about stuff like doing that. We should be thinking about saving material for use at a later date. But as a scientist, I would say, let's go one step further and decide, is that the right community we should be trying to move that person towards? Or should we use the opportunity of a short-term disruption like antibiotics to introduce some other things or do some reconfiguring at the same time?
So if they're healthy, I offer them their own poop. If they're unhealthy, I offer them my poop.
That could be an option.
Or somebody else who's got no gut problems. Yeah. I mean, it's a problem we have. I mean, in medicine, we have this Hippocratic oath of first do no harm, but I think that's a bunch of crap since we're talking about crap today, because if I wanted to first do no harm, I would never do anything, period, because there's always harm and risk associated with everything we do. So it's do what's probably more beneficial than harmful. That's the decision we have to make with every patient in front of us. So this is something that I'm going to continue digging into, because I really do think this could be a valuable thing. But I'm with you. I would love really clear, definitive data on this, on autologous fecal microbiota transplant, which is what that's called. We do have great data on treating C. diff with FMT and other disorders that is helpful for as well. I think this could be one where there's some benefit. And I mean, I feel like if it's your own stool, it's a pretty low risk intervention. But I wish I had more data.
So there's one other challenge with that I forgot to mention, and it's that the collection, the processing, the preparation and administration of the sample is one of the technically challenging aspects of this. And many of the microbes that we know to be some of the most beneficial members of the microbial community are extremely sensitive to oxygen exposure. Most of the microbes that live in our gut that we label as the good guys that are there are obligate anaerobes, meaning that they can't grow or even die in the presence of normal room levels of oxygen. So collection of a sample, how we store it, how we prepare it, and how we get it back into the person is absolutely critical to making sure that those really oxygen sensitive microbes don't die in the process. And it raises the question that if we're not careful about how we process a sample or how we deliver it, we might be losing a lot of the good guys in the transfer. And what we give to that patient after the fact might actually end up be something very different than what we collected from them because of the sensitivity of those microbes along the way. That's a technical challenge that we face a lot in the laboratory, working with the germ-free model of how do we get stool from a patient into a mouse and do it efficiently enough that we get the right community establishing in the mouse.
Yeah, interesting. So I mentioned the effects of the microbiome on anxiety and depression. We know the increased risk of those when we disrupt someone's microbiome. We know the microbiome's role in producing serotonin, which everyone understands, I think, at this point, its role in depression and mood. And we're going to talk specifically in another podcast with a psychiatrist about this issue, how the microbiome affects anxiety, depression, Alzheimer's disease, autism spectrum disorder, all of these, what we think about as brain disorders, which are really linked to the microbiome. Do you have any kind of thoughts or anything you want to share on that to kind of tease that future episode?
Yeah, absolutely. The gut-brain axis is an area of emerging importance in microbiome, and I'm really excited to see where that part of this field goes. Some really fascinating work has come out in the last 3 to 5 years, tying together microbial community structures in the gut and behavioral and psychiatric effects in the brain and in the CNS. And it's a level of communication and connection that I don't think anyone really predicted, but we're seeing really strong associations. Sarkis Mazmanian's lab out at Caltech is doing some really wild stuff, understanding the effects of the microbial community on autism spectrum disorder and also on Parkinson's disease. And we're seeing really, really interesting interplay between the types of microbes that are in the gut signaling through the vagal nerve and other neurologic pathways, and then that having effects on behavior and anxiety levels. And it opens up a whole different door for how we may approach treating some of these conditions as opposed to the canonical neurotransmitter inhibitors and other pharmacological treatment methodologies. Maybe we should be looking towards the microbes as a way to do this for us.
And Dr. Lynn has done some amazing work on Alzheimer's disease and the microbiome. We're going to be interviewing her soon on a podcast related to that. And our Wild Health Director of Oral Health, Dr. Stein Van Arsdal, has done some amazing work on kind of oral health in general, periodontal disease and its relationship to Alzheimer's disease. So we really want to dig into those topics. I know, though, that you don't have a lot of time. I really just wanted to introduce people to you today, just so that you all know, if you're listening, that if you've got really intense, deep questions about the microbiome, send them to us now instead of us and interpreting the data and the literature on this. We have someone who's been intimately involved in that research and is much more qualified than us to answer those questions. So Eric, thank you so much for joining us today. And can you answer some questions later if some people send in some questions to us?
Yeah, absolutely. I'd be happy to.
All right. There's your commitment. Send some questions. We'll get the answers to those. And tune in next time. The next podcast, we're talking to the Chief Science Officer of Viome on exactly how much you can test right now and how much directional information you can take and act on from those tests.
We hope you like listening to the podcast as much as we like making it. If you think it's valuable, share it with somebody. Send it to someone you think would really benefit and leave us a 5-star review on iTunes. Those reviews really, really help us out. Find us on Facebook and follow us on Instagram at WildHealthMD. If you didn't like, well, why are you still listening? Email us and let us know what we can do better. But the best way you can thank us for our time is to leave a review and share. Until next time.
Taglioli. It's like ravioli.